just want to take a bit of time to draw our attention back to the syllabus and kind of get a frame of reference in our mind about the pace that we are taking and when to anticipate the first exam. So I'll switch over to the computer, and we see here's today, Monday the 24th, and then Friday the 228th of January. It'll come faster than you think. Obviously, I got a little typo there. We begin discussion of gametogenesis today. We will discuss the male gametogenesis first, and then on Wednesday we'll pick up with Lecture 4, actually probably complete Lecture 3, and move into Lecture 4 on the description of the female reproductive system and a focus on the human menstrual cycle. And then we will proceed into Lecture 5, looking at fertilization and straight through into the developmental series of events week by week. February 2nd is the census date. So if you withdraw by that day, if you determine that you need to, then there will be no grade showing up on your record. Then we begin to look at specific developmental features, looking at particular systems and the structures that are developing in relationship to that. So we'll start with an embryo that's just a 3-cell layer thick disc or plate, and then eventually it's going to be growing so fast and it's constrained, kind of like a purse string around it. It has to curl and fold and do all these things, and as it does so, it'll create body cavities, and other events will occur like the emergence of the circulatory system. So we'll proceed along those lines, and then in the other parts of the semester, and after looking at the placenta and labor and delivery, we'll begin to look at each organ system, how it develops. So we'll do a global picture of everything first to get a feel for the whole developmental series, build a bit of strength in our nomenclature to start with, and then after we consider the births, then we begin with cavities again, basically a review but with more detail. Then we proceed through the development of the head and neck, and certainly start with the systems. So you see exam 2 will be March 7th. There's quite a stretch between exams. Exam 1 is on Monday, February 14th. Again, that's when it'll be posted, and then you'll see on the web CT when those will be okay. So the exams icon isn't showing yet. I thought I had it there already. Okay. I'll get that posted, and then when you are able to see the exams, you'll see the dates associated with them. And the other thing is, I had a pretty busy weekend, and I didn't get a chance to generate the links for the PDF files. I'm sorry about that. I'll try to work on those tonight and have them up by Wednesday. So there's still plenty of time to work on reviews without having to print massive numbers of the lectures yet. Another point, the calendar shows the scheduled events as well. So the Monday, the February 14th is the, just double checking that I got the right date on there. So happy Valentine's Day. I had a chance to look at the emails this morning, but I didn't get a chance to respond. I'll work on those as the day goes by. Well, let's switch over to the lecture then and discuss gametogenesis. So we have a better feeling for meiosis, even though I kind of got my wires twisted toward the end of the last lecture. We do understand that there is the 24 chromosomes in the diploid, and the haploid gametes have only 23 unduplicated chromosomes. They don't have their homologue with them. And there's a major distinction between mitosis and the first meiotic division in that the homologues separate from each other through the nuclear division events. It's not until meiosis II that the sister chromatids of the duplicated chromosomes separate into the final 4 haploid cells. So meiosis II is a little bit more like mitosis, except we don't have the homologues together, but the event of splitting the sister chromatids of the duplicated chromosome is the same. So with that background then, we'll proceed to discuss the development of the gametes. So we will, of course, be considering the histology of the gamete-producing organs, and then when we talk about the reproductive endocrinology, we'll revisit these organs because these are mixed organs. They are endocrine tissue as well as producing the gametes. So the same for the female. We'll review not extensively, but at least the relevant histology and anatomy for understanding how the gametes are produced. So first then, we consider the male gamete development, spermatogenesis, and we know that it begins at puberty. So now, well, first let's remind ourselves that developmental biology spans conception, or gametogenesis basically, but more specifically conception to death. Right? So there's, what's the term for the biological process of maturation from age 10 to 20 or so? I'm sorry, I can't hear you. Somebody's phone is... Puberty? Is that what you were saying? Were you answering?
Well, puberty or okay. Are you talking about for both males and females? Or?
Yes.
Oh. Okay.
So the biological term is puberty, and then what's the term for the psychosocial development during that time? Adolescence. Okay. So puberty is a phase, childhood, infancy, neonate, all those terms. Adult, midlife, early senescence, death, all those are included in developmental biology. So we're studying human embryology, a particular stage of that, and we will obviously proceed to the study of the fetus as well, up to the point of birth. So it is during puberty, of course, that the gametogenesis begins in the male gonads. However, up to that point, there's still many developmental events that occur. So we'll see a slide of an early fetal testy and then an adult testy. So the histology would look very different at different phases of development throughout life. So we think then first of the reserve cell, the spermatogonia. So these are pools of diploid cells, and they will undergo the mitosis. We said before that if the reserve cells undergo mitosis, they must have one of those 2 daughter cells remain as a reserve cell. So there's something a bit different about that cell, and then the other daughter cell will go on to differentiate. It may still have some limited capacity for mitosis, or meiosis in this case, right? A strange division than other cells. But ultimately, that one daughter cell that goes on to differentiate goes to its functional senescence and death as well. So the spermatogonia represent the reserve cells with a significant mitotic capability. That generates that daughter cell 2, if you will, or there's a daughter cell A, daughter cell B nomenclature. The differentiating daughter cell, if you will, is now called a primary spermatocyte. So one spermatogonia doesn't produce 2 primary spermatocytes. It produces another spermatogonia and a primary spermatocyte. That reserves some modified division capability and goes through meiosis 1, as we had talked about. That's the reduction division. And then the secondary spermatocytes, the 2 secondary spermatocytes produced by that one primary spermatocyte. So here we have a case of differentiation. We're not developing another reserve cell. We split into 2 daughter cells that are identical and follow a similar lineage of cell division and differentiation. Are they truly identical? No, because there's a little mixing of the DNA that we had talked about, the synapsis and the recombination of the DNA. And they each have one homologue of a duplicated chromosome. Then the secondary spermatocytes will be the haploid cells, and they will undergo meiosis 2. They are haploid in that they they have oops, did I mess it up again? These are diploid here, then they become the haploid. And each contains the duplicated chromosome. And then the secondary spermatocytes will produce the spermatotids that are haploid and have the unduplicated chromosome without the homologue. The spermatids are the final stage. There will be 4 of those, right? And then they will undergo a development. So we are talking about spermatogenesis as a 2-phase process. There's the meiotic events to produce the spermatids, and then we'll discuss spermiogenesis, the final phase of maturation of the spermatids to become fully functional motile sperm. We'll spend a bit of time on this slide to generate an overview, then there will be a significant amount of review as we progress. We, of course, are looking at this panel on the left at a sagittal section through a testy, and we see the thick connective tissue surrounding it. Anybody know what that's called? Tunica albuginea, with the septa, the connective tissue separating it into chambers, and then within each chamber, of course, are the seminiferous tubules, and this will be our focus on the development. So here we see a portion of a seminiferous tubule pulled from that. They are highly coiled structures, and the sperm cells will develop continuously from puberty when it's established through the senescent phase of adult malehood, and out through the entire length of the seminiferous tubule, there will be the same events occurring as we see in a cross section in this next panel at a higher magnification. So we'll review the anatomy of this as we proceed, so we can see that along the length of each of the seminiferous tubules, the sperm are being produced. The production is from periphery to center. The center is the lumen of the tubule. Typically, reserve cells. Now, if we enlarge a segment of one of these tubules, we see the periphery on the top in this middle panel, and the central, the lumen, at the bottom of this panel. At the periphery, we see that we are at the deepest layer of the epithelial lining. So these are epithelial cells. They are gamete-producing epithelial cells. We know there are two types, the sustentacular cell or the gamete-producing cells, the sperm-producing cells. So the apical portion of these sustentacular or Sertoli cells is toward the lumen, and the base is toward the connective tissue basement membrane. So the sustentacular cells are attached to the basement membrane. All the hormones that come in to affect these cells come through that basement membrane. The vasculature is outside, as we'll see and we proceed. So this orientation to the cell is extremely important. So what we're beginning to build now are approaches to establish criteria to orient to magnification, base of the cell, apex of the cell, and various characteristic features that identify a cell or tissue for you visually. So we see the nucleus of the sustentacular cell here. So obviously sustentacular, it sustains the development, and we'll see that. So very often, virtually all cases of epithelial linings, the reserve cells are going to be at the basal layer of the epithelial cells. Now, epithelia can be single cell layer thick or multiple cell layer thick, but obviously it's only one cell layer thick. The reserve cells are right there. That's the deepest layer. It's only one cell layer thick. So it stands true in virtually all cases. So we orient to the periphery. We know that these spermatogonia are going to be distributed throughout there. So when we proceed to look at some actual histological slides, we should be able to look and see nuclear appearances consistent with mitosis. Maybe see a metaphase or an anaphase or a late prophase, something like that. Not that we're going to try to tell exactly what it is. Metaphase would be an easy one. But we'll just try to note the difference in the nuclei when they are in interface or early prophase versus any of the later stages. So the progression of the development of the sperm, the differentiating cells, the primary spermatocytes, are going to be deepest, secondary next as we approach the apex of the cell, and the spermatids at the apical membrane intricately associated with it. The sustentacular cell cytoplasm surrounds the developing cells, the spermatocytes, and of course encases the reserve cells, the spermatogonia, at the base. As well, the spermatids, when they are undergoing spermiogenesis at the apical membrane, have intimate association with the sustentacular cell cytoplasm, actually engulfed within it, until finally they are released into the lumen and migrate through the lumen of the seminiferous tubules through a series of other tubules, the terms of which we don't need to focus on in this class, and then finally into the epididymis. The epididymis, as we will see, is the final site of spermiogenesis. The epididymis has head, body, and tail. If you sampled sperm from the head, they would be less mature, not the least mature, because the least mature would be within the seminiferous tubule. The body, there would be more motile sperm, and in the tail of the epididymis would be the fully mature, fully motile sperm capable of conceiving or fertilizing an egg. So the notation to point out on the right-hand side here, the spermatogonium are diploid. They proliferate by mitosis. Not really proliferate, but I like to use the term proliferate because they produce more of themselves, but they just replace themselves, start with one and end up with one. So you don't end up with 10 times the number of spermatogonia. They replace themselves and they produce one new cell that will go on to differentiate. Then the primary spermatocytes are 2N, and they will undergo the first meiotic division and will produce the secondary spermatocytes, as we said, undergoing the second meiotic division, until finally we have the second set of haploid cells, the spermatids, that go on to complete their differentiation called spermiogenesis, until ultimately the mature sperm is produced. So with the long tail, the mitochondria and the midpiece, the neck region having the centrioles, and then of course the head containing the nucleus and the acrosome, and we'll discuss those in a bit as we proceed. So let's look at this again in detail. The seminiferous tubule is the focus of our attention, so we're going to zoom in on this box. First one set of views of the human testes to represent that developmental process before and after puberty that I had mentioned. The early testes in a newborn has testes cords. So these are the seminiferous tubules beginning to develop from the mesenchyme in that area, the differentiating tissue. It's undifferentiated and some differentiated cells that are within there. It's very important for us to begin building in our mind this idea of cellular differentiation. What's the difference between totipotent, pluripotent, multipotent, tetrapotent, tripotent, dipotent? So what does that mean? It's a cell that's developing along its lineage, or a series of cells along its lineage, until there's a point where it can either become this kind of cell or that kind of cell, or finally unipotent. It can only it's a reserve cell that can only produce those kinds of cells, and those cells go down the final lineage of differentiation. So in a developmental course like this, it's extremely important to understand this lineage of potential for cells to produce other things. And this is very important, obviously, in society today, because we hear so much about the stem cells. Well, basically, they're all stem cells up to the point of the reserve cell, and then some of the reserve cells still have some capabilities that can be played with, so they may even function as stem cells. So there's some of those reserve cells exist within us, so they're able to get some stem cells from adults. So it's important to keep all of that in mind. But the farther down the lineage you go, the more differentiated that cell is that has mitotic capabilities. So there are less tissues that you might be able to produce from it, unless you can de-differentiate back to a more multipotent series in the event. You can be limited in what tissues you might, or what cell types you might produce from that stem cell. So a true stem cell from an early zygote obviously is totipotent, can become any type of cell in an organism. So it's important to keep that in mind. So out here, this just kind of looks like a loose connective tissue, but there's a lot of differentiating cells proceeding to produce all the different cellular layers involved in the ornate structure of the gonads. So these are obviously the early developing seminiferous tubules, and they are referred to as testis cords. In the adult, we see that in this cross-section at probably relatively similar magnification, there are many of these cut in all different planes. We got this one obliquely in a turn, kind of as if it was cut along this angle here. Others that are almost completely cross-sectional, as if we had a 90 degrees to the long axis. And we note that there's always the space outside of, between two of them, or among all of the seminiferous tubules. So this is interstitial space. It's the connective tissue matrix of the stroma of the testi. It's a term to work on Stroma is the connective tissue structure that supports things, and so that would be like all the structure to the building. That's the connective tissue stroma. Then we're the parenchyma. We're the functional cells that reside within that connective tissue stroma. So it's important to understand organs from that sense. So the neurovascular bundles and the lymphatics are all going to be distributed out here in this space outside of and around and among all of the seminiferous tubules that we see. So again, these are the efforts to get oriented to a histological view. What cells do we find out here in this interstitial space? The Sertoli cells. The interstitial cells, no, of Leydig. You drew me in with that, didn't you? So the interstitial cells are found here of Leydig, and they, of course, are responsible for producing the testosterone. We'll see more of that as we proceed. So obviously, from a neonate stage to full maturity, post-pubescent, a great series of maturational events will occur. This is a high magnification, I don't know what type of a stain it is, of a mature seminiferous tubule. We orient to the lumen and to the periphery. We see the connective tissue stained. What are these empty circular spaces out here in the interstitial compartment? Vasculature, right? Capillaries, arterial side of the capillary bed, venial side. We got to get blood in and blood out if we're going to produce testosterone in the nearby interstitial cells of Leydig. We have to deliver the hormone that's going to stimulate it, and we have to wash away the steroid when it is secreted. So therefore, any hormone delivered here that is going to impact the Sertoli cells, the sustentacular cells, has to diffuse from the capillary through the interstitial space out here in this interstitial compartment, and we're just using that. It's not really the best definition for interstitial, but we'll just go with it since we link it to interstitial cells there. It has to diffuse through all of that, not get degraded, get through the connective tissue basement membrane of the seminiferous tubule, and then bounce around in the interstitial space in here until it bumps into its receptor on the Sertoli cell. Of course, the steroid, the testosterone, diffuses rather freely through all of that. The hormone being delivered to the Sertoli cell, the FSH, is a peptide, so it has to bump its way along a little differently. So you have to picture that. The interstitial cells of Leydig being stimulated by LH, testosterone is produced, it diffuses everywhere, so it gets over to the Sertoli cell, and then coming from the capillary, the FSH has to bounce around until it gets through and bumps into its receptor. So I obviously am very oriented toward the microanatomy of events and being able to visualize how things are happening. We often are presented this type of material to think that, oh, this hormone's produced in the pituitary, and then it's got in its mind, I'm going to the Sertoli cell and nothing's going to stop me. Obviously, we just bathe the interstitial fluid compartment, delivered by the plasma, and it goes everywhere. And when it's at a place where there's a target cell expressing the receptor, and it happens to bump into it just right, then it can have its effect. So that's why I play these games of bouncing the mouse around and stuff. Then we can imagine almost immediately that the base of the Sertoli cells is in the periphery. There are primary spermatogonia, there are spermatogonia there that are going to be triggered to divide. Let's see if we can see. Here's a dividing cell there. We can see some of the chromosomes there. You can see some clustered here. Should be able to pick out some out in here where the secondary spermatocytes are residing. Can't really see a lot of them. And then, of course, this is the apical cytoplasm here with the spermatids developing, and we see the tails cut in section within the lumen. So this is an illustration of what we were just looking at. And again, we're building our ability to visualize and recognize these types of things in actual specimens. So this is a single cross-section through a Sertoli cell. We orient to the connective tissue basement membrane. This is the basal layer of the epithelium, basal membrane of the Sertoli cell, the sustentacular cell, and its apical membrane. So there's a difference between a basement membrane and the basal layer of the cytoplasmic membrane of the cell. The basal layer of the cytoplasmic membrane of this cell is going to have the receptors for FSH. The FSH is going to diffuse out of a capillary out in that interstitial space that we're allowing ourselves to call it, the connective tissue stroma of the organ, diffuse through and finally get to its receptor on the Sertoli cell. And then here are all of the sperm-producing cells. So we see that there's some artistic liberty taken by the artist in this illustration. We have noted in the histological sample that we just looked at, there isn't this nice fine clustering and columnar sequencing of things. They're just all over the place. And again, we note the interstitial cells of Leydig outside of the basement membrane of the seminiferous tubules. LH coming through, binding to the receptor, sustaining the synthesis of testosterone that diffuses freely everywhere. You got blood flow, you selectively wash it away. And then the diffusion in to the receptors within the sustentacular cell. So one final view of this sustentacular cell and its association with the production of the sperm, the spermatogonium, 46 chromosomes, primary spermatocyte, 46 chromosomes, but they are, they are duplicated chromosomes. We have the full homolog set, and then the secondary spermatocyte has, there would be 2 of these produced, has haploid number, but they are duplicated chromosomes connected at the centromere, and then they split to produce the spermatid. Spermatid starts with the nucleus and a full spherical cytoplasm, undergoes a series of changes, dramatic changes, sheds cytoplasm even, until finally it is the morphologically mature or complete sperm, not functionally mature, and would be released into the lumen to travel on to the epididymis. And that process from spermatid to sperm, of course, is spermiogenesis. So some questions inserted relevant to that. So again, note the distinction between spermatogenesis and spermiogenesis. Spermiogenesis is the final stage of spermatogenesis. Meiosis is the first stage. So again, that's reviewed here. I'm just going to point out a couple of other terms on this slide, because it is covering the same information, but it allows us to distinguish spermatogenesis as having 2 phases, meiosis and spermiogenesis. And the final part of spermiogenesis is to produce the mature, fully motile spermatozoa within the tail of the epididymis. The other terminology on this slide shows the basal compartment of this epithelial layer and the adluminal compartment. So we remember the terms ad and ab, the prefixes AD and AB. If you are abducted, you are taken away. If you move your arm away from the midline, you abduct it. You bring it back toward the proximity of the midline of the body. That is adduction. If you take a piece of adhesive tape, it adheres to something. So AD means that it's close, right? So adluminal compartment is closer to the lumen of the seminiferous tubule. The basal compartment is at the base of the epithelial layer at the periphery of the seminiferous tubule. Here's where they introduced the terminology. The spermatogonium producing daughter cell. The daughter cell type A spermatogonium remains in the basal lamina as a precursor cell. And daughter cell type B spermatogonium becomes the primary spermatocyte that will go on to produce 2 secondary, and then 2 secondaries produce ultimately 4. So we start with 1 primary spermatocyte and end up with 4 early spermatids. The process of spermiogenesis occurs at that end stage of the meiosis when the haploid spermatids are produced. There's a series of events that occur. I scanned in the figure legend right with this, so it accompanies it. And we can see, just to highlight this and proceed, there's very important events that occur. First off, we notice a change in shape, a rearrangement of intracellular structures. So immediately, what subcellular organelle do we suspect is involved? Shape and intracellular movement of organelles, cytoskeleton. We know from the previous slide that the centrioles are in the neck region here, and then the tail becomes the motile part. So centriole and motile extension of the cell, subcellular organelle, cytoskeleton. So the cytoskeletal proteins exist to change the shape and help to shed the cytoplasm, and then of course there's a significant production of all of the microtubules that end up within the centrioles and the tail. Anybody remember the arrangement? It's 9-2 microtubules, that type of thing. So it's very similar to a cilium in that sense. What else do we note that is occurring? There are 2 other significant events here. Obviously, they're labeling the Golgi and the mitochondria. So what's happening to the mitochondria? They're migrating toward the neck region, and then ultimately end up right in this first part, just after the neck. I forgot, it was labeled on the previous slide. I forget what it was called. So all the mitochondria are located there. Why do you think that would be a good place to have them? Because you require all the ATP for the motility, right? So got to pump out a lot of ATP to keep the centrioles spinning and the tail moving. Then the other structure that's labeled, obviously, is the Golgi region. So it undergoes a tremendous rearrangement from the typical multi-layers of pita bread is the way that I usually envision it. And somehow they all just fuse together into one big giant pita bread and have a lot of different proteins contained within it, and then just drapes over the top of the nucleus and picks up a new name called the acrosome. We see the shedding of residual cytoplasm as this proceeds as well. So the acrosome is going to contain some very important proteins. They almost make it appear that there's no longer any cytoplasmic membrane on the outside of the the acrosome. Since we have the document camera set up, let's try to envision what would really be there with a drawing. So if this is the nucleus, right, trying to do the 2 layers for the nuclear envelope with the nuclear pores, then there has to be somewhere out here the cytoplasmic membrane. So draped over this nucleus is this structure produced by the Golgi, the acrosome. So we may lose the cytoplasm here ultimately, so that the cytoplasmic membrane becomes very tightly adherent to this outer layer of the acrosome. The reason that this is important is that there are still membrane proteins in the cytoplasmic membrane of the nucleus that are important for the binding to the cells that surround the ovulated ovum, mature ovum. Anybody remember the term for those collection of cells that surround the ovulated ovum? Forms a crown that radiates. Corona radiata. So again, we try to remind ourselves that this thin cytoplasmic layer stays very close by. That just means we've got a couple lipid bilayer membranes pushed together. And then ultimately, that acrosome is just going to open up and dump all of those molecules onto or into the corona radiata to push its way through and ultimately get through a carbohydrate polymer layer, the zona pellucida, surrounding the egg. So it needs those enzymes within the acrosome to do that. So here's a question, spermiogenesis. So be sure you focus on that and try to find the actual definition for it. It may seem like a slide describing something, this process from spermatid to mature spermatozoa, but these actually would be the choices for a multiple choice question. So obviously the formation of a functional sperm by the stripping away of superfluous cytoplasm would be what we're focusing on there. Okay, anybody want to tell me what this is? It's obviously the cross sections of adjacent cylindrical tubules. Well, orient to the basement membrane of this circular cylindrical structure. You see any signs of the spermatogonia, any arrangement of cells so that there's some separation of nuclei. All these nuclei look almost exactly the same, don't they? What are these things sticking off up here? What's the term for long cytoplasmic extensions off the apical membrane of a cell? Well, cilia. But these don't move. They don't have the microtubules in them. So they are stereocilia. Sterocilia. And then here in the center is a clump of maturing spermatids, or sperm. So this is the epididymis. And looks like nobody's moving in there, so it must be the head, right? Okay, bad joke. So this is the connective tissue stroma surrounding them, a lot of fibroblasts out here, blood vessels, arteries, veins, capillaries, lymphatics, and so on and so forth. Okay, so you've already learned enough or developed enough skills and sharp visual acuity to ask certain questions. These are cylindrical structures cut in cross-section. I see 3 of them there. I see the area outside of them, the epithelial layer, the lumen, the apex of the membrane, the base of the membrane. These don't look like the spermatid tails sticking out like we saw on any other slide, no matter how it was illustrated or stained. So what cells, actually these are the only cells in the body that have stereocilia, so as soon as you see those and they kind of stick together like that, you know that it's epididymis. Nothing else will look like that. And then the maturing sperm with the processing of the tissue will clump together within the lumen. So this is the epididymis where development will occur. The spermatogenesis continuing. The spermatozoa, the haploid cells, undergo the cytoplasmic changes to become the mature sperm, the spermiogenesis. So the head contains the DNA, the tail, it was centerpiece, wasn't it? That was the term where the mitochondria was located in the tail. The centrioles just at the neck, if you will, and the mitochondria are there as well. Oh, microtubules. This uppercase MT is microtubules, excuse me. So this is describing that whole thing that what I've called the neck, the centerpiece, and the tail, just kind of including them all in the tail. No harm in that. And then the acrosome contains enzymes essential for the sperm that contact the corona radiata to migrate deeper in and race to become the winner. So there's obviously a male bias in a lot of the reproductive biology. And we always seem to picture the sperm just racing each other, trying to get there as fast as they can and stick to the corona radiata and be the first one to contact the zona pellucida and get through it and contact the membrane of the ovum and be the winner. Is it that passive from the side of the ovum? No, they're actually molecules secreted by the ovum, by the ovulated cells, let's call them the corona radiata plus the ovum, that attract the sperm. And then there's actually a selection process. The first sperm to get to the zona pellucida and through it may not be the one selected by the ovum for the fertilization. So there's a lot more biology known. I guess once female scientists got involved and discovered it, guys just didn't think to look for it, right? So the stereotypical scientist, I guess. And then the ejaculate contains 100 million sperm, and this concentration or total number, of course, is indicative of degrees of fertility. So infertility is less than 50 million in a single ejaculate, and sterility is less than 25 million in an ejaculate. Now we see the middle piece of the tail and the neck of the tail and the principal piece of the tail and the end piece of the tail. And I really couldn't quite figure out why in the text they show this little one and then this. Still, if that was actual size, that'd be significant. Okay, so here's another slide. And question 1 on this slide is, what is this structure? Anyone? Obviously, a cylindrical tubular structure, cut and cross section, lumen, basal, and the periphery. Looks like all the nuclei are pretty much the same in appearance, clustered toward the basal cytoplasm of the epithelial cell layer. From the apical cytoplasm, there's these long things sticking out. Lumen's empty, but that's okay. So it is the epididymis. What process occurs here? Now, spermiogenesis, right? The final stages of spermiogenesis where motility is achieved. Okay, here's another histological slide. So first, we don't really see these are cross sections of a cylindrical structure, but we see the arching appearance of this layer of cells here. Looks like a little bit of connective tissue stroma in the organ. And another similar structure nearby there. We see cells marked at the edge of that connective tissue, obviously a basement membrane holding epithelia in place. And as we look around, we see, here's where we can see strong evidence of cells dividing, undergoing mitosis. So what is the structure? Seminiferous tubule, and the process that occurs here? Spermatogenesis. Good. This is what we were looking at, basically. So like this illustration, it shows the capillary bed in this area, and we remind ourselves that hormones from the pituitary diffuse down their concentration gradient into the interstitial fluid compartment of the connective tissue stroma, and are randomly diffusing away, bumping into cells. When the LH binds to the LH receptor on the interstitial cells of Leydig, activate certain things and sustain the synthesis of testosterone, right? And the FSH bounces around, as does everything else in the blood, glucose and everything, bouncing around, trying to work its way through this connective tissue basement membrane of the seminiferous tubule, finally bumps into the FSH receptor and sustains certain events in the sustentacular cells such that they sustain the growth of the production of the gametes in the male. And then the testosterone, as it is released, diffuses away freely throughout this area, washed away in the capillary blood toward the venous side, but also getting to the sustentacular cells and binding to the androgen receptor there and producing certain effects. Okay, what structure is this? Is it low magnification or high magnification? Moderately high, I'd say. Lowest magnification is where you'd see the cross-section of the whole organ, or enough of the organ to say I know what organ this is. It's moderately high. What are these dots? What's that bigger, darker thing with euchromatin and heterochromatin? I guess that gives it away, doesn't it? This is a nucleus. These are nuclei. What are these things coming out here? Those are the tails of the spermatids, right? So these are spermatids. So this structure is seminiferous tubule, the apical membrane, if you want to get really particular about it. And what cell type has the smallest diameter? Well, if you obviously recognize that this is the apical membrane of the seminiferous tubule, then it'd have to be the spermatids that are the cells of the smallest diameter. Because here's the nucleus of the other cell that we can see, probably a primary spermatocyte somehow nearby up there. And this is the margin of its cytoplasm all around out here. This might be a secondary spermatocyte there, or maybe an early spermatid. I guess way up here, this probably would be a secondary spermatocyte. And then this would be a spermatid, right? So you have to be able to begin putting all these together. Okay, what about this structure? Definitely sperm around here, spermatids. Now look at the cells. All the cells are uniform. So you don't always get that nice, perfect section through the epithelial layer, but obviously this is cells very close together, very little connective tissue in between them. So this is an epithelial cell there. I've got epithelial cells and sperm. Very likely it's the seminiferous tubule. Can't really see all of the stereocilia in this stain, but if you look, you see a few of them. And the process that occurs here, again, is the spermiogenesis. Okay, we'll begin on Wednesday with the female gametogenesis. I appreciate the few extra minutes and your time and attention. I hope you all had a good weekend.